So my name is David Peeler. I'm a research fellow working across labs in the departments of materials, bioengineering, and infectious disease here at Imperial College London. And my work is on self-amplifying RNA delivery. So we focus on how nanomaterials can shape how RNA is expressed in the body and where and for how long and what cells it interacts with. And the goal of our work is to make better, safer vaccines for infectious diseases, although I also work in cancer research. My work has centered around immune interactions with materials. And so central to that work is understanding the immune system. And so mice are a central model in all of our work because you need an intact, complete immune system to understand the outcomes of all of the possible interactions.
Yeah, super nice.
The immune system is not like a typical organ. You could think of it more like the brain or the central nervous system, where it's throughout the entire body, and it has many different parts that need to communicate and actually are totally mobile throughout the body. So it would be very, very difficult to recapitulate interactions between the two arms of the immune system that we care most about in vaccines and in all therapeutics, which are the innate and the adaptive immune system. And those are two full, complete repertoires of cells that need to be able to come together and interact in different locations in the body. And so my work actually directly studies what is going on in those different locations in living mice. We can anesthetize them just like people are anesthetized for procedures at a hospital. And because we work with self-amplifying RNA, which replicates inside of cells to make copies of itself and continually produce protein, which is actually the same protein that's in the butt of fireflies, which makes them glow. So that's how we can see that the protein is expressed. And then take pictures of them to see where, for example, in their muscle, the protein is being expressed and to what extent. So from that we can get like a curve of how long and how sustained the expression is, and also understand where roughly in the body that RNA is being expressed. And importantly, the nanomaterials that we make can dramatically change both how long, how much, and where that RNA is expressed, which directly affects how the vaccines work. The 3 R's are really important in our research. There's a reduction, refinement, and replacement, and we use all 3 of those in our practice. For example, the imaging that I was just talking about, being longitudinal, we don't have to sacrifice mice at each time point. We can track a single mouse over time. Another strategy that we can benefit from is the fact that mice have 2 legs, so we can give 2 different vaccines in each leg. One vaccine just encoding that protein that we can easily see the expression of. And then in the other leg, we can encode like a flu antigen, so like hemagglutinin. Unfortunately, mice are not a perfect model for our work because, as you can probably imagine, mice are different from people. And so the innate immune system of a mouse has evolved slightly differently from how humans developed much later. And so because the innate responses are so essential to ultimately vaccine success, because it's the starting point of everything, we also have replaced mice in some cases or augmented mouse research with the usage of human skin from various plastic surgeries. Because skin is actually the biggest immune organ in the body and contains a lot of interesting innate immune cells that we can analyze. So that's been really rewarding work that I've been doing in Professor Robin Shattuck's group, and that his group has a long history of using like human explant models, which are totally free and perhaps more clinically relevant than a mouse model for very specific studies. Again, we can't just do clinical trials all the time, although that would be great. Probably it's more ethical to use mice for the adaptive immune studies. It is filling up though. So I've been lucky to get some support from Imperial through their 3 R's grant scheme. And in particular, it was a good opportunity for me to do some extra experiments to refine the technique. And so while a small grant, it was very helpful as a visibility booster and also as a training tool to help me share this technique with both animal facility staff as well as students in the department.